Hi, my name is Taeyoung Kim. I'm graduate student at Busan National University. Today, I'm going to talk about multimodal transformer-based deep neural network for determining burst positive structural variation course. In genomics, structural variations are the variation in structure of an organism's chromosome. Structural variation is generally defined as genomic mutations bigger than 50 base pair size. It could occur in DNA or RNA and mutated like insertion, deletion, and duplication, and so on. Moreover, some structural variations are associated with disease like cancer or genetic disorders. It tends to be nothing general case, but if some critical region is mutated, it could harm health. So it is important to get high confidence structural variations. In this study of Cameron, we could show the rates of first positive structural variations. The best score on the NA12878 is Gridis, which also has first discovery rates over 20%. Moreover, they tend to have a very strong trade-off between recall and precision. So precision dramatically drops when we try to get high recall score. In the study of Kosugi, we also shown the F1 score. Some cores get high F1 score in deletion, but the other types of structure variation have low F1 score, so it needs to refine first positive structure variations that are recorded by these tools for more high confidence. So we first determine the first positive structure variations. I used Manta, RomPy, and Dervis, which are generally used when calling structure variations, and acquire gold standard dataset integrated by Thousand Genomes project. In the next step, we randomly sampled at 50, 20, and 10 coverage respectively from list of the 300 coverage dataset, and then create alignment data using BWA MEM program. These alignment data would be used for getting structure variation information, and next stage is detecting a set of candidate structure variation use the cores. These structure variations are determined by gold standard data. Label the data as true positive when a candidate structure variation overweighs 90% with structure variation in the gold standard. If it's not, determined as first positive structural variation. After determining true and false positive structural variations, we make deep learning model for predicting false positive. We build multimodal transformer introduced by Taeyoung in 2019. We embedded the reference genome sequence that observed the region of structural variation. Sequence data is embedded as vector per base pair and is converted as pole dimension. If they have soft masked sequence, just multiply 0.3. These data are used as text modality. Next, we convert alignment data to image suggested by Ryu 2021. This method extracts spirit read depths, and read pairs using same tools and convert this data as like figure 3. In figure 3, read depth represents as red area and spirit read represents as purple area, and read pair represents as yellow area. These data are used as image modality and convert alignment data to signal format. Like image modality, extract speed read and read depths and read pairs as per base pair, and they are normalized from 0 and 1. These data are used as signal modality. After pre-processing work, we put these modalities in 1D convergent neural network layers. For running transformer, we need to match the dimension of these modalities, so we resize dimension of each modality using 1D convolution neural network. First, We pad each data to maintain data size after putting in 1D CNN and calculate 1D CNN for each vector depending on kernel size. So the dimension of output data is judged by the number of kernels. Each modality has same dimension after calculating 1D CNN. In this part, we put these modalities into cross-modal transformer. In this layer, embedded modality A is considered as source modality and B is considered as target modality. This source modality is calculated query with query weight and target modality is calculated as key and value with key weight and value weight before putting in multi-head attention. Then query, key, and value are calculated using scaled dot products parallelly and take skip connection as adding data in the layer norm. And by passing through the position-wise bit for the layer, the attention of the data is aligned and derived. This course repeats n times. After that, we could get fused pictures from B to A. After calculating cross-modal transformer, we put data in self-attention transformer. This layer does the same thing as the layer in the previous step, except that it uses as the same data to generate a query key value and uses a scaled dot product. This slide shows the performance with F1 score. Deep SB filter is the base model to compare our model results. No filtered results show the lowest F1 score, and our model has best F1 score in every coverage. In this study, we propose a method to determine burst positive SV. Our model shows lower number of burst positive SV than other cases, and high and low coverage also show good results. And we can show multimodal running take advantage of running low-level representation of SV. But due to premature convergence of learning, overfitting more increase and the tendency of misfit, the test data increase. If we improve this part, we expect to get more high performance. Thank you for listening my presentation.